An overview of exercise 8, activity 4 of your Physio X lab. We're talking about the digestion of fats. So you will recall, right? Fats are hydrophobic. They clump together in the gut. That means that with a big glob, little surface area relative to the volume. So the enzymes don't have much access to the fats. Which enzymes, you ask? Lipase. So lipase are the enzymes that are coming from your pancreas and being released into the small intestine. And their job is to take a triglyceride and break it down into a monoglyceride and 2 fatty acids. So let's go back to like week 2 or 3 of class. And remember that when we eat fats, what we're eating are triglycerides. So think about your favorite fatty food. Maybe it's ice cream, maybe it's an avocado, maybe it's avocado ice cream, it's a thing. So the triglyceride is, there are 3 carbons, that's the glycerol group, and then there are 3 fatty tails on that triglyceride. That molecule is too big for you to absorb through your small intestines, that simple column epithelium. So what the lipase does is it's going to break some covalent bonds, and here and here. And what that does is it converts our triglyceride into a monoglyceride. So 1, 2, 3 for our glycerol, and then we're going to leave this 1 fatty acid tail. That's our monoglyceride. And now, since we've broken those bonds with the lipase, then we have these 2 free-floating fatty acids. Diagrammed so well. Turns out that is small enough that it can get into the cell. Note that we are making more acids. So one of the ways, well, the way this lab works is we're going to indirectly measure the increase in fatty acids, because what will it do to our pH? Boop, right? More acids mean more acidic environment. So we're going to see a decrease in the pH if we successfully digest triglyceride into monoglyceride and 2 fatty acids. Wait, there's more. What's this step about? Let's talk about bile. So bile is produced by the liver, stored in the gallbladder until it gets released. More on that in your recorded lecture. But once it gets released, that gallbladder is a little muscular sac, right? And it can squirt. And when it does that, it's going to release that bile into the small intestines. Same time, your pancreas here is going to release lipase, the enzymes that are breaking triglycerides into monoglycerides and fatty acids, into the small intestines as well. So what is this bile doing? Well, it's going to break up or emulsify those fats into smaller droplets. Well, I know, there's always more to it, isn't there? First of all, as you eat, those fats are hydrophobic, and so they want to congregate together. Fortunately for you, your small intestines are moving all the time. They do this thing called segmentation, which basically means they're just kind of squishing or milking. Instead of milking in one direction, which would be peristalsis, it's kind of squishing stuff back together. And so the chyme, or the digesting food, is kind of swishing around. And what that does is it takes a big glob of fat and breaks it up into smaller pieces, emulsification. But what helps maintain those small droplets as bile, that it's going to surround. Once that glob gets broken up, then the bile salts and the lecithin in that bile is going to surround those little fat droplets and keep them small, so they have a high surface area relative to their volume. And now the lipase can do its work of having lots of access to the triglycerides and do the digestion. That's the background. Here's what the lab looks like. You will start this way, and then we'll have you move some of those test tubes into the test tube rack. Voila. And once you do that, then you're going to get this chart that is going to have you set up a bunch of treatments. Now let's go back here. What have you got here? You've got lipase. Okay, so what's that? Yeah, that's your enzyme. So that's actually going to do the reaction. I'm going to see if I can improve my handwriting here. Get a different pen. Blame the pen. Okay, vegetable oil. What's that? What do we need that for? That's our fat, right? So that is our fat source. That's where the triglyceride is. And so that is our substrate, the reactant that's going to go into the active site of our enzyme, the lipase. Bile salts. Yeah, that should increase our surface area to volume through emulsification. So if we add bile salts, what would you expect to happen to our chemical reaction rate? Is it going to increase or decrease? Okay, that's the top shelf. What's on the bottom shelf? Well, you can see there are pH buffers. You've learned about buffers. What's a buffer? It's going to resist pH change. Yeah, so what we're interested in this experiment is looking at what's the effect of different pHs on our lipase. So at what pH is it most effective? Where do you have a pH of 2? In your stomach, right? And then you have a pH around 7 in the small intestines, and then, well, let's just throw in pH 9 and see what happens. So you're part of this experiment, you're looking at the effects of different pHs on the enzyme. And then the deionized water, what's that there for? Well, you're also going to be setting up some controls. And the deionized water is used to make sure that you have the same volume or the same concentration of solutes in all of your 6 test tubes. So you will set up your test tubes, and you can either do one test tube at a time, or you can be like, oh, lipase, I need that in tube 1, in tube 2, in tube 3, and whoops, not in tube 4, in tube 5, and tube 6. So you can grab the dropper and drop appropriately. Whatever organizational tactics works for you. And then at the end, it should look something like this. You'll incubate them, and then you end up putting them in this assay cabinet. And what the assay cabinet is basically a pH meter. And so you're going to be recording the pH of each one of these tubes, and then comparing what you find as far as how active the lipase is. And how are you going to measure the lipase activity? Right. Lipase makes monoglycerides and fatty acids. So the more fatty acids you make, the lower the pH. I also want you to think about which one of these tubes are serving as controls, and what do you think the role of each of those controls are? All right.